yeah, like I said, please, stop me or interrupt if you have any questions about any of this. This is joint work with Alex Williams, Matt Golub, Surya Ganguly, and David Cicillo. And again, thanks for the invitation. Okay, so probably like many of you, I'm very much interested in connections between deep learning and neuroscience. I think, deep networks are like a really interesting tool that I think are going to shape how we think about both, like how we think about questions in neuroscience and how we go about understanding the kind of computations implemented in neural systems. And if you think about it, I mean, the goals of these kind of fields are quite similar. In particular, we're studying networks which have similar goals in that we want these networks to, in the case of deep learning, we're trying to train networks to have flexible, intelligent behavior across a variety of tasks. And in biological networks, we're kind of given these networks through, studying animals that have been shaped over the course of evolution and their lifetimes. But in both cases, we're trying to study how networks solve interesting tasks. And I think what's quite surprising is there's been a recent plethora of work which has shown that deep learning models trained to solve particular tasks learn very similar algorithms as what we see in biology. So just to give you some kind of examples of this, there's a whole host of papers, especially in recent years, where people have basically trained a deep network to solve a task and shown that the kind of internal either representations or computations or even algorithm used by the deep network is very similar. It matches quantitatively the representations and algorithms we see in biological networks that we think are responsible for kind of solving the same task. So this kind of slide just gives you like an overview of a lot of the kind of diversity of studies like this. And these kinds of studies, cross both different brain areas, different kind of levels of the neural hierarchy, as well as like many different kinds of animal species. And so I think in this particular work, we were kind of motivated by, trying to think about why, we were kind of wondering how surprised should we be when we see all of these different kinds of similarities. In particular, it seems quite surprising because these networks have very similar representations. But if you actually look in what they're built, what the kind of underlying mechanisms that we use to build these networks, they're very composed of very drastically different elements. So on one hand, you have, biological mechanisms, which involve biological neurons, cellular mechanisms, molecular mechanisms, and so on. And on the other hand, we have these artificial neurons, which we kind of dream up, which typically involve composing simple computations like linear filtering and pointwise nonlinearities and things like that. So the kind of central question guiding this particular paper was, when you train networks to perform the same task, why should we expect artificial and biological networks to be similar at all, given differences, these drastic differences in underlying mechanism? And in particular, can we kind of understand, as you change these three things that I've colored here, as you change, say, the tasks that the networks are trained on, as you change the kind of metric or measure that you use to compute similarity, or as you change, say, underlying mechanisms, when and how should we expect these things to be similar? I think it's important for a number of reasons. In particular, if you're kind of at the interface of neuroscience and machine learning, and you're trying to carry out this enterprise where you train neural networks to solve tasks and then compare the internal representations to biology, you have to make a lot of choices, right? When you train your neural network, you have to choose, what kind of neural network you want to train, all of the different architectural parameters and training parameters and so on and so forth. And so, if we see kind of some match between biology and an artificial neural network, we would want to know, how robust are those similarities to, for example, changes in those particular choices you made when you were designing your artificial network. Okay, so that's going to be the central question. Before I dive into that, I just want to touch on a brief aside, which is a lot of this talk is going to be talking about universality, and I want to do a brief aside on I think one of the more beautiful examples of universality in dynamical systems, which comes from the logistic map. And before I kind of dive into this, I'm just going to do like a brief 5-minute overview maybe. But before I dive into this, I think the lessons we're going to learn from studying universality in these kinds of simple dynamical systems maybe will translate or be like informative for the lessons that I think and that we would like to have or take for this kind of enterprise that I outlined before. Okay, so diving into the logistic map and universality. So this is the logistic map. It's a very simple one-dimensional map. So basically you map points X to the at iteration N to the next iteration through this simple function. And R is like a little parameter of the system that you can tune. And as you tune R, you get very different kinds of behavior in the system. So for example, what this plot on the left is showing is just iterating this map for different, for 63 time steps. So you can think about this as like running this little dynamical system. And so the animation is going to play, it's going to show what happens as you increase this parameter R. So, for small values of R, for example, shown here in blue, you get basically a single fixed point in the dynamics. Everything just converges to this one point. But then as R crosses a particular point, I think it's around 3, you'll see now what starts to emerge is a stable cycle. So now instead of having a single global fixed point, you have this 2 cycle, but it's called a 2 cycle because it basically iterates between or oscillates between 2 points. And on the right side is just showing the Fourier spectrum of this signal. So you can see there's a single peak here at the Nyquist frequency, just showing that there's a periodicity in the signal. And what's interesting is you keep increasing R, all of a sudden you'll see what pops out is now you have a 4 cycle. So basically there's 2 peaks in the Fourier spectrum, and the map is, or the system oscillates between 4 different values. And then as you keep iterating R, all of a sudden now the map is iterating between 8 different values. So you have an 8 cycle. So this is what we call period doubling. Basically, as I change this parameter R, the system goes through repeated bifurcations where it goes from a stable 2 cycle to a 4 cycle to an 8 cycle and so on. And then at some point there's a transition to chaos, and then that's as you keep increasing R. Okay, so that's period doubling, and the behavior of this system was studied by Mitchell Feigenbaum in the 20th century, and he found a really interesting property of this system. So on the right side, what I'm showing is what we call the bifurcation diagram for this system. So on the x-axis is this order or the control parameter R, which is this quantity right here, and then the y-axis is just showing the kind of stable, the limiting behavior of the system. So again, for small values of R, you just have a single fixed point. So that's indicated by just a single point over here. And then you go through a bifurcation where now all of a sudden you have a stable 2 cycle in the system, and then a stable 4 cycle and so on. And then out here you get chaotic behavior. Now, what Feigenbaum found is there's actually a surprising limit that governs this kind of behavior, or that you can kind of extract from this system. And that limit is the ratio of how quickly it takes to period double. So basically, you think about this as like the length for the first period doubling. This is the length for the second period doubling. This blue bar is now the length for the next one, and so on and so forth. And what he showed was there's this constant known as Feigenbaum's constant, which is the limit of these successive ratios. And that limit ends up being just a particular number, 4.669 or whatever. Okay, so that's the Feigenbaum constant, the logistic map, but kind of nothing I've said so far really speaks to anything, universal or anything like that. I think the real surprise or kind of cool thing about this work is he shows, or, we've seen that this constant shows up regardless, in many different cases, regardless of what the actual underlying function you use or what underlying map you iterate. So for example, on the left is the logistic map. And, as you keep iterating, the limit of this ratio goes to Feigenbaum's constant. Here's a, different map. It's a slightly different equation. Again, you get Feigenbaum's constant out. Here, in this case, this is like from here's a complex number. This is from the Mandelbrot set. And again, you get Feigenbaum's constant. So what's cool or what's interesting or intriguing about this is you get aspects of the behavior of these systems, which are in some sense universal, across different kind of underlying computations, right? So regardless of which of these sets of equations you iterate, you kind of get this universal behavior. So that's a very brief overview of universality, but I think that's the kind of, I think when we started doing this work, the kind of dream scenario for us was that, eventually maybe we'll get to some understanding where we understand both, what are the kind of universal aspects we see when we study computational systems, in this case, dynamical systems, regardless of kind of the underlying, in this case, the actual equations underlying the system. But in our case, again, we're going to change the actual equations underlying the system as well. People, since study this and understand like why you get this particular behavior and like what properties of the system needs to have in order to get this and so on and so forth. So that's the kind of understanding that we would like to have. Okay, so now coming back to our question, again, just to reiterate, we're trying to understand when trained to perform the same task. Why should we expect artificial and biological networks to be similar, given that there's such drastic differences in these underlying mechanisms? And so in this study, to take a first stab at that, what we did was basically just an empirical study, and we're only going to study artificial networks. And again, I think I'm probably preaching to the choir here, but just to motivate artificial neural networks in neuroscience, there's a lot of advantages of using artificial neural networks as kind of model systems in neuroscience. In particular, you can easily train artificial networks to accomplish tasks kind of analogous to those we used to study biological networks in animals. And then once you have a trained network, it's very easy to, inspect or probe or dissect what the artificial network is doing. You have much better experimental access to these networks compared to something like a biological network, where you have to do, very involved experiments to kind of probe the network. And finally, we can do very large numbers of these kind of in silico studies by training and analyzing artificial networks on a scale which is just impractical or impossible to do with biological networks. So for those reasons, we're going to kind of focus on just artificial networks here. So basically, just to make a long story short, what we're going to do is train artificial networks with different mechanisms on different tasks, compare them using different kinds of similarity measures, and then see, when do all these different networks, what properties look similar, what properties look different across these different tasks. So first, I'll kind of briefly talk about the different mechanisms or underlying mechanisms that we varied. So we basically trained a large number, like thousands of recurrent neural networks, and we varied properties of these networks. So we varied in particular the recurrent architecture. So like we tried vanilla RNN, GRUs or gated recurrent units, LSTMs. And these are basically different, specifications for the underlying equations governing the recurrent neural network. So we varied those. We also varied the activation function used in the network. We tried tanh and rectified linear units. And we also, we varied things like L2 regularization as well. Okay, so then given this now, we have this kind of huge body of networks which have been trained on tasks. I'll explain the tasks in a minute. The next thing we also did was compare a couple of different ways of assessing how similar or different two networks are, right? So, I think another kind of big question in neuroscience right now is how do you, compare kind of two different biological or neural networks. And so basically we use two different measures of network similarity. One is, we think about this, the first one is like kind of measuring the representations of these networks or the geometry of the representations. So this uses what's called canonical correlation analysis. So it's looking for a linear projection of the activities of the units in two different networks that maximizes their correlation. So if two networks have very similar activity, then you can imagine, a linear projection they'll be well correlated with some arbitrary linear projection. And so that's kind of what this similarity measure is assessing. The other kind of similarity metric we looked at is a little bit kind of task specific, but what this goes into is what we do is we analyze, so these are all recurrent neural networks, so we go through and we analyze the dynamics of these networks, and we do that by studying fixed points of the dynamics and linearization and corresponding the dynamics around these approximate fixed points. So I'll explain that kind of as we go task by task. But this is kind of very different from the previous similarity measure because the kind of fixed points and dynamics, the way we're going to, quantify them, don't actually depend on the particular geometry of the network activity. And so instead, this is kind of like extracting like we think of it as like almost like a topological structure of what the network is doing. And you'll maybe see more clearly what I mean by that. Okay, and then finally, we did all of this on 3 different tasks. We wanted to make sure that, whatever conclusions we draw maybe aren't particular to one particular task. So the 3 tasks that we studied, I'll kind of walk through the results for each, but there's a like discrete memory task, which we call the flip-flop task, a simple pattern generation task, and a contextual integration task. So yeah, again, so I'm just going to walk through the results task by task. So first, the discrete memory task. So let me just explain what the task is. The task is from one of David's previous papers, but it's a very simple task. Basically, the network gets some input, which is given by this like black trace here, and the correct output for this network is just has to remember the previous input. So the inputs are either pulses up or down, so -1, +1. And if the output was -1, then the network just has to stay at -1. And if the output flips, if the input is a positive pulse, then the network has to flip and retain the positive pulse. So it's just remembering the most recent input that came in. And so this is for one input, but we can kind of make the task more complicated by having multiple inputs, and the network has to generate the appropriate output for each one of them. So in this case, we have 3 of these inputs. So we call it a 3-bit flip-flop task. Okay, so the first thing we did was, again, we trained a bunch of networks to solve the task. All these tasks are quite simple, so all the networks solve them to basically perfect accuracy. And so now what we're going to do is we're going to compare, we're going to ask how similar are these networks. So the first thing we did was look at what we call representational similarity. So that's using this canonical correlation analysis. So what I'm showing here is going to be a distance matrix, where every point in the distance matrix is the distance between 2 networks. So how do we compute distance? Well, basically we take the network activity for one network and another network. We compute the canonical correlation analysis. So that gives us a set of correlation coefficients. So if all of those correlation coefficients are 1, then the networks are all like perfectly identical. And then if they're 0, then they're extremely dissimilar. So we just quantify distance as 1 minus the average of these CCA correlation coefficients. So now what I'm plotting here is, all of these pairwise distances for these different networks. And to kind of simplify things, I've just sub-selected all of, I think in this case, it was just the TANH networks. But here you can see what happens is you vary the different architectures. So you have the vanilla RNNs, the LSTMs, these update gate RNNs, and these GRUs. And in particular, black colors here indicate very similar networks, so the distance is 0, and white indicates very different networks. And basically, the thing to take away is you can see it's very strong block diagonal structure in this similarity matrix, right? So, for example, all of these vanilla networks look very similar to one another, and all of these gated recurrent units look very similar to one another. And there's a little bit of more structure for these networks, but they also have this kind of block diagonal structure. I think this is varying. We're also varying L2 maybe in here, which is why you have this additional structure. Anyway, the point is that these, look block diagonal. And another way of kind of seeing this just visually is we can reduce this matrix. We can embed this distance matrix using something like, so before I get to the embedding, this is again the same picture now where I'm just showing the distance matrix for TANH and ReLU networks. Here I think I've again sub-selected out just the vanilla RNN. But again, you see very strong block diagonal structure. So you can see, across all of these different TANH networks that we train with different random seeds, you can see they all look similar, and all these different ReLU networks look similar, again, according to this kind of similarity measure. And so if you then take these distance matrices, you can kind of embed them in 2-dimensional space using multidimensional scaling, which just gives a kind of, I think, a nice qualitative picture of what's going on, which is basically that all of these networks cluster, right? So using this similarity measure as a distance measure, and then trying to embed these networks in a 2-dimensional space, which preserves that distance, reveals clusters, right? So you get the vanilla networks over here, the GRUs over here, so on and so forth. And then over here, again, the ReLU networks look clustered and the TANH networks look clustered. And so, okay, so why is this interesting or important? I think that the important bit is now, let's say you had a biological system trained to solve the same task. Now, you're only going to have one or a very small number of examples of that biological system, but you can imagine doing the same kind of distance computation between that system or that network and all these other networks. And when you do that, that biological neural network is going to be some point in this space. And now the interesting thing is, depending on whether you happen to choose a vanilla network or an update gate network or a GRU cell or whatever, you might conclude that your network is either very similar to the brain or very different, just depending on these kind of architectural choices. Okay, so from this perspective, things look quite individualistic, right? Depending on these little choices, networks can look very, very different. If you actually study these networks, so there's aspects which do look quite universal, and that's the network dynamics. So what I'm showing here is a low-dimensional projection of the network hidden state. So these are, we varied the number of hidden units, I think like 64, 128, 256 units. But basically, for all of these networks, if you project their hidden states into the, you do principal components analysis, so you look at the dimensions that capture most of the variance, project into that space. For this task, these networks, the trajectories look like they form this kind of cube structure. So all these different gray lines are like different trajectories of the system as they're processing different inputs. But basically what they do is they kind of ping around these different corners of this cube. And again, just to show that I'm not like, this captures most of the behavior, you can see across all of these networks, this is like a histogram of how many PCA components you need to explain 90% of the variance in hidden states. And for most networks, it's around 3 or 4. Okay, so gives you kind of a qualitative picture of the dynamics. This is a little more quantitative. So here, this is, again, it's a little bit task specific, but what we're doing is we're showing the fixed points of the network dynamics. And the fixed points, I think, really show you how the system's solving the task. So we can numerically find approximate fixed points of the dynamics, which I'm kind of skipping over, but basically when you do that, what you find is all of these little dots. And this is for an example network, but basically you see this structure across all the networks. And so if you look at the dots, what you find is basically there's 8 stable fixed points, which are these black dots here, and they form the corners of the cube. And then in between them, you have saddle points. So in between any pair of stable fixed points along the edge, you have a saddle point, which is indicated by these red points. These are saddle points with one unstable direction. And then on the face of each side of the cube, you have a green point, which is a 2 saddle that has 2 unstable directions. And then right in the middle, you have a 3 saddle. Basically, the way we think about this is the networks kind of set up these dynamics where basically spread out these stable fixed points. The stable fixed points are like memories. And what happens is particular inputs now push the system over into a different attractor basin and then get attracted to the appropriate fixed point to remember that particular memory. And so if you remember, this is a binary, each input is binary, so you have to remember up or down, and there's 3 of them. So you have 2 to the 3 possible states or memories that you need to keep track of. Hence, you have 8, 2 to 3, or 8 possible of these stable fixed points. Okay, so we can kind of analyze this task, and we see that basically we see the same dynamical structure across all of these networks. So even though the representations look kind of different, if you do like CCA to quantify the representations, when you do fixed point analysis and look at the dynamics, those look very similar. So we wanted a way just of quantifying this similarly to how we kind of quantified the CCA structure. And so the way we did that was kind of the simplest thing we could think of. Basically, we just form a little graph of which fixed points have dynamics which lead to other fixed points. We generate a graph of all of these fixed points. And what we do is we basically take, so this is part a little bit task specific, but what we do is for a given approximate fixed point, we initialize the system around that fixed point and see where the dynamics go. And so you can imagine if we initialize from all these stable fixed points, the dynamics just return to the stable fixed points. But if we initialize from these saddle points, the dynamics will then go to different fixed points. And so that forms what we call this like fixed point adjacency graph. So it's just kind of schematized here. On the left axis is like where we kind of start the system. So we're starting it very close to these approximate fixed points. And on the x-axis is like where the system ends up. So this black bar are all the stable fixed points. So if you start at the stable fixed point, you end at the same stable fixed point. That's why you see the identity matrix up here. Down here, if you start at these unstable fixed points, you always end up at the stable fixed points, which is why this first kind of column is the only one with color. Everything out here is all zeros. But this kind of shows how the unstable fixed points work and then how the other unstable fixed points work. The reason we did this is basically this kind of gives you, it's like a way of quantifying kind of like a dynamical portrait of the system. And the reason we did that is because if you look at it, this kind of portrait across all of these different network types, you see a very similar portrait. So this is what I mean by the dynamics across these networks look very similar. And so then we just take these adjacency matrices, and then we can vectorize them and use them to compute a distance between these networks, which is now like a distance between like kind of the dynamical structure of these networks. And so again, if we do that multidimensional scaling and embed these in 2 dimensions, now you see basically all these networks lie on top of each other, which is just another way of saying that all of their dynamical portraits from the perspective of these approximate fixed points look quite similar. Okay, so that was the story for this 3-bit flip-flop task. Basically, the story is going to be the same for the next 2 tasks. So I'll kind of walk through those next. So the next task is this pattern generation task. So think of this as kind of like a motor task almost. So basically, the network gets an input, which is just a step input. And the overall value of that input, so the how strong that input is, tells the network what frequency it has to generate as an output. So basically, if it gets an input of 1, then it has to generate a sinusoid with a 1 hertz frequency. And if it gets an input of 3, then it has to generate a 3 hertz, then an input of 5, and it has to generate 5 hertz. And when we train the network, we just vary this input between 1 and 5, and then train it to generate the appropriate sine wave. So it's kind of generating these little patterns. Now, again, if we can look at the representations of these networks. So again, what I'm showing here is a low dimensional projection of the network hidden states using principal components analysis. This is like PC1, 2, and 3. And each color here represents a different frequency input. So, the yellow ones are the high frequency inputs, the purple ones are the low frequency inputs, and you can see the network is generating the appropriate oscillations for these different frequencies. And again, if you look at this across a bunch of different networks, both TANH and ReLU, you see that the networks all, one, they also solve the task, and 2, if you actually look at the, kind of shapes of these oscillations, they actually kind of look a little bit different, right? So for example, like this one looks a little more squashed, and so on and so forth. But they're all low dimensional, right? So again, all of these need roughly three dimensions to capture 90% of the variance. Okay, so if you again compare these network representations using CCA, I'm skipping over the part where we generate the distance matrix, collapse it using multidimensional scaling. So these are just the cluster plots using multidimensional scaling. But basically, you can see the networks kind of cluster, right? In particular, like the ReLU and TANHs look separated, and these different cell architectures look separated. And so what that tells us is that maybe if you were to use, again, CCA to assess how different these networks are, you might conclude that networks look different using CCA. But again, if we go back and look at like what are the actual dynamics of these networks, that actually looks somewhat universal. So let me explain how the dynamics work here. So in this case, there's basically a number of different approximate fixed points, but for a given input, there's only one fixed point. So here I'm coloring these fixed points by the corresponding input to the network. So, the purple are the low frequency inputs and the yellow is the highest frequency input. So the networks learn to kind of set up an input dependent fixed point for each of these different possible inputs. Now, why is that useful? Well, for a given fixed point, it's very easy to generate sinusoids just by having a particular oscillation of a particular frequency for that fixed point. And so that's kind of what I'm showing here. What this plot is a plot of the eigenvalues of the recurrent dynamics for each of these fixed points. So this is like the linearized dynamics. Again, these are all nonlinear dynamical systems. What we do is we just, for a given fixed point, compute the Jacobian, the linearized approximation of the dynamics. And these are the eigenvalues of that Jacobian in the complex plane, so the real axis and the imaginary axis. And this dashed line is the unit circle. And the unit circle is interesting because that's the, points on the unit circle will have stable, points within the unit circle have stable dynamics. And on the unit circle, in particular, if you're off on the imaginary axis, will have stable oscillations. And so you can see here, again, these points are all colored by the corresponding fixed points. So you can see these purple points have a low frequency oscillation, and then as you increase the input frequency, oscillation of the corresponding frequency gets faster and faster and faster. And so what seems to be universal in these networks is they all kind of use these input dependent fixed points and approximate linear dynamics to solve the task. So just to drill into that a little bit more, what we're showing here is like the actual input frequency that we fed to the network and the kind of predicted linear frequency if you just assume the whole system was a linear dynamical system, computed its Jacobian, and computed the linear frequency from the eigenvalue of the recurrent Jacobian. And basically, they all kind of match up, right? You see that they all have this linear relationship where the higher the input frequency, the faster the corresponding eigenvalue. And so again, we came up with a simple way of quantifying this topology. Basically, we did another like little chain where we build a graph of the neighboring fixed points for any given input frequency. And then when you use that graph and do, again, this multidimensional scaling, you see that all the networks kind of fall on top of one another. So even though the actual, these wiggles, these oscillations look different from network to network in the actual recurrent units, they all solve the task by using, in this case, input-dependent fixed points. And that's the part that's universal. Okay. Final task is a contextual integration task. So I'll go through this kind of quickly. It's again going to be the same story. These tasks are heavily inspired by like decision-making tasks in neuroscience, where the network sees kind of noisy sensory evidence and a particular context. And so depending on this context bit, the network has to either integrate, in this case, the blue evidence or the black evidence, and then reach a particular output. And again, just to make a long story short, when you train networks on this task and assess their similarity using CCA, you again find these very clear clusters. So it looks like all of these networks are kind of doing something different when solving this task. But if you actually look at, again, the dynamics of these networks, so here what these pictures are showing is a very simple probe where I just feed in, instead of white noise input, just feed in a constant step input to the network. And so the expected output is just a linear ramp, just to very clearly show the dynamics. Basically, all these systems kind of start at some point, but basically integrate evidence along what in these are shown by these gray points, which are approximate fixed points. And these approximate fixed points form a line in each of these cases. And so basically, all these networks use like integration along a line attractor to solve this task. And here I'm just showing one line attractor, but there's actually two because there's two different context inputs, and so that's why, anyways, the network learns two separate line attractors in this case. And again, if you then, the fact that they all use line attractors is the universal bit. So if we then go in and try and quantify that, again, we see that all the networks kind of lie on top of each other. There is this kind of interesting difference in this case between ReLU and TANH, which you can kind of see here, which is that for whatever reason, the ReLU networks kind of have this bend or kink in their approximate line attractors, which the TANH networks don't have, and that's kind of showing up in how we quantified these similarities. Okay, so conclusions. I think from this little set of empirical studies, what we found was that representational geometry, which we again computed via CCA, is quite different between different models. So it's indicative of these like task irrelevant architecture choices. By task irrelevant architecture choices, I mean things like, what recurrent cell do you use, what nonlinearity you use, and so on. But there's aspects, in particular the fixed point topology and the corresponding dynamics, which seem more universal. These are insensitive to these architectural details. Basically, all the networks use kind of what I consider as like the same algorithm to solve these tasks. And so that's kind of what we think of as like universality. And I think the general point is that, these issues are really important if we're going to continue to use deep learning to understand neurobiological data, right? We need to know, what similarity measures to use, what architectural details are important or not important. When can we trust, statements about how similar deep networks are to biological networks and so on. So I think this is just a first step in that direction. Again, it's all empirical, and so we just, all we can make statements about are the findings for these particular tasks and similarity measures. But at least I think we did a very large number of networks, and again, across all three tasks, we found that the fixed points of dynamics are universal, whereas the representations looked more individualistic. So that's the kind of story for this paper. I just want to say briefly, just kind of touch on like related and additional work we're doing at Google. So this is kind of beyond the scope of just neuroscience, but I'm really in particular interested in this kind of procedure of reverse engineering neural networks. So taking a trained neural network and trying to understand, what algorithms the network's using to solve particular tasks. And so we've kind of done that in a few cases now. There's a really interesting story with how recurrent networks solve a natural language task, which is sentiment analysis. And what's interesting is they seem to solve it very much how we see integration dynamics happen in neuroscience. So they use like a one-dimensional line attractor and integration of evidence to kind of solve these sentiment analysis tasks. Again, in language processing, we've looked at things like document classification and other classification tasks. And there we also see really interesting geometric structures which seem universal across different tasks. So in this case, you see, depending on how many classes that the network has to classify, so how many different kinds of documents the network has to classify, it learns either line attractor dynamics or plane attractor dynamics. And the number of classes, the geometry of these attractors is related to kind of the number of classes. And then, outside of neuroscience, I think we're also applying this to other kinds of algorithms. So this one is maybe a little bit out of scope, but it's trying to reverse engineer how recurrent network trained to do optimization works. And there, again, using tools that we use in neuroscience. It's all like principal components analysis, linearized dynamics. Those are the basic tools. You can really, I'm not going to really explain this, but you can kind of start to really understand how these trained systems work. And I think that's super important. I think the tools that we use to understand artificial neural networks are very likely to be helpful for understanding biological networks. And I think both the actual like algorithms that these networks discover might also be algorithms that we might see in biological networks. So I think from Google, it's very easy for us to do these kinds of reverse engineering studies. And I think that's kind of like, I think an exciting direction I'm looking forward to pursuing. So anyways, thanks everyone for listening. Happy to take questions. I know I went a little long. In particular, Alex Williams and David Zasilo. Alex was like a co-author on all of this work, and David, I think, was really the driving force behind a lot of the like dynamical systems analyses and so on and so forth. And yeah, please contact me with any questions. Happy to take questions now as well.